Ernst. I'm faculty at UCLA. I'm going to be talking about computational approaches for understanding the non-coding human genome. So protein coding portion of the genome covers about 1% of the genome, and the vast majority is not protein coding. And it's important to understand this part of the genome as it's been seen from disease mapping studies such as genome-wide association studies that about on the order of 90% of the genome is these variants identified by genome-wide association studies are falling into these non-protein coding regions of the genome, and many are likely having regulatory effects. So it's important that we have an understanding of what mutations in this part of the genome would affect. So in my work, I've looked at this problem from a number of different angles. One is using epigenomic data. Another is leveraging high throughput functional testing data, such as from massively parallel reporter assays. And a third strategy is based on comparative genomics analysis. This first part I'm going to skip today because it will be covered in my tutorial talk next Friday at 1:30 p.m. So you'll hear about some of the methods that I've developed and related work in the field for taking epigenomic data and annotating the genome based on that. But today I'm going to focus on these last two parts, the work on massively parallel reporter assays and some work on comparative genomics conservation state analysis. So with the massively parallel reporter assay data, this technology has sort of been used to follow up on this question that based on epigenomic data, we have the ability to identify thousands of candidate regulatory regions, and you'll find out next Friday specifically how one can predict those specific regions. But we now have, in any given cell type, it's quite easy to generate thousands of predictions of regions that are potential regulatory regions. But we would want to have some ability to test them in high throughput, but not only to test them, but to dissect them at a high resolution, to identify where the activating and repressive nucleotides are. And we're going to focus on doing this independent of sequence information. There's a lot of strategies leveraging sequence information, but we're trying to come up with an orthogonal annotation and to differentiate activation from repression. And we're trying to do this for thousands of regions at once. And the enabling technology for this work is something called a massively parallel reporter assay. So this is specific technology that I'm showing you was developed by a collaborator of mine, Tarjei Mikkelsen, and a number of other groups developed closely related technologies at the same time. And how this works is one can synthesize DNA sequences that one is interested in testing for the ability to drive gene expression. So in this specific design here, thousands or tens of thousands of DNA sequences can be synthesized and placed upstream of a reporter gene, and then you have a unique barcode in the 3' UTR. So if that sequence you've synthesized is able to turn on gene expression, then you would get that barcode sequence. So you can have a count of the number of copies of each barcode you had in the original DNA, and then you can, after transfection experiment, count how many of those barcodes you have at the mRNA level. So this gives you a very quantitative readout for thousands or tens of thousands of sequences that you have complete control on how they're designed. I mean, one limitation is they're about 145 base pairs in length, and this assay can also be noisy. So oftentimes people put multiple different barcodes for the same sequence they're synthesizing. So in the original application of this technology, what Tarjei did was he took two well-known enhancers and his collaborators, and they did a systematic mutagenesis of each nucleotide, mutating it at in each possible way with multiple different barcodes to control for noise. But because they were devoting so many different barcodes to and testing each variant in each possible way, they were limited to only testing 2 enhancers. So by this mutagenesis, they are able to identify activating and repressive nucleotides, but there's this overall limitation on the number of sequences you can reasonably synthesize and transfect. They could only test a couple enhancers. In follow-up work led by my colleague Puya Kradpour, we tested more than 2,000 enhancers predicted based on the epigenomic data, and also did targeted mutations of regulatory motif sequences. So these are patterns in the DNA that the transcription factors recognize, and did targeted testing of motifs that we thought are important. And this was largely a confirmatory study. It wasn't allowing us to discover de novo activating and repressive nucleotides, but allowed us to test a large number of distinct enhancers. But we're interested in the question, could we effectively combine the elements of these 2 studies to, in one experiment, to test thousands of regulatory regions and at the same time gain access to very high resolution information as to where the activating and repressive nucleotides are, and differentiate their direction of effect. And we're doing this also without knowing the precise 145 base pairs to test. And one of the challenges here is that the more reporter sequences we devote to interrogating any one location, the fewer locations we can test overall. So this is where we need to be sort of smart about how we actually allocate these reporter sequences to each location to get a lot of information out of a small number of reporter sequences per location. And in order to do this, it turns out that there's a fairly simple design, but it becomes quite powerful if you think about it the right way computationally. So what we do is we take locations we're interested in testing and we tile them with 31 reporter sequences. Each reporter sequence is 145 base pairs in length. So we cover 295 base pairs in total, and these reporter sequences are in 5 base pair increments. This has a number of advantages. One is that in a single experiment, we can test now close to 8,000 regulatory regions. Experimentally, we were able to scale this up to testing close to 244,000 unique barcodes or sequences in a single experiment. So by only devoting 31 reporter sequences per location, we could test close to 8,000 regions. And we didn't need to know precisely which 145 base pairs to test. So based on the chromatin data, you can often be confident at a 200 to 300 base pair resolution where the relevant regulatory site is, but it's difficult to be confident on a specific 145 base pairs. And then we're only devoting 1 barcode per single position, but we're able to control for noise because there's a lot of redundancy between closely related positions. But that is also by reasoning about these offset positions and the relative activity is what gives us information to recover at high resolution regulatory bases. And I'll explain to you how we do this computationally soon. But before I get to that, just to briefly say a bit about the specific data that we tested. So this was, all the experimental work was done by my collaborator Tarjei Mikkelson's group. And what we ended up doing was we took locations that were all centered on DNase I hypersensitive sites. So these are regions of open, accessible DNA. So there's an assay that can specifically find these regions. And they were selected to be in different chromatin states. And you'll hear about this next week. But just sort of very briefly, based on different types of epigenetic marks and their combinatorial and spatial patterns, we can partition the genome into these different types of chromatin states. So one of the objectives of the study was to understand how these different states would have different activity in this assay. And then we did a stratified random sampling to have coverage of DNase I hypersensitive sites in each of these chromatin states. We tested in 2 different cell types, with 2 different types of promoters, a minimal and an SV40 promoter. We did it with 2 sets of sequences, and everything was done in replicate. So 16 experiments in total, yeah. There was, I think, a couple hundred base pair spacer region. I would have to check exactly. Yeah, it was put on a plasmid where there's a fixed distance. So first, to gain some intuition about how to think about this type of data. This is 295 nucleotides of DNA sequence that we're interested in testing. And then what you see below it are 145 base pair subsequences of that DNA. And gray represents basal expression. Now suppose there was a sequence right here which was sufficient to turn on gene regulation or activate the gene. What we'd expect is these subset of tiles to have a higher expression and the remaining to still be at basal expression level. Now suppose there was an important activating sequence right here that was sufficient to turn on the gene. We'd expect these tiles to have above average expression and just these ends to be at basal. And if we had a sequence right here which was sufficient to repress the gene below basal expression, we would expect to see these subsets of tiles have depleted expression and the remaining to be at basal. This is a very, that was just sort of a toy example. In practice, it won't be that deterministic. So what we need was a probabilistic modeling approach that would go from input data like this to an overall inference of the regulatory activity at each position along the region. So these are two actual input examples. So yellow corresponds to higher expression, blue lower expression, white is missing data. And what you're seeing here in blue is the inference based on the minimal promoter, in green SV40, and then black the combined. So you can think of those as two replicates. So the method's effectively making a prediction that there's a strong activating sequence concentrated over here, while there's a strong repressive sequence over here. And we can compare this to predictions of regulatory sites based on motif information, which looks at the DNA sequence. And specifically, I'm showing you here predictions based on the centipede method, which is based on motif information and some other information, including DNA's footprints. And they're making a prediction in this specific cell type we're looking at that there was a motifs right here and motifs right here. And you can see it lines up quite nicely with the predicted activating and repressive sites. But we have sort of two added advantages over this motif-based prediction. One is that we can differentiate activation from repression. And two, we could potentially find regulatory sites that don't correspond to any specific sequence pattern, because we're making our predictions independent of the DNA sequence, the specific pattern in it. So now to go to the actual probabilistic model and how we went from the inferences, from the data to the inferences. So just to remind yourself here, the input data we have is activity of these 145 base pair sequences. And what we would like to do is have inferences at sort of close to this 5 base pair offset resolution, which we don't have directly, but we want to infer by reasoning about the relative activity of these various tiles. So in our modeling approach, what we do is we have an observation Y1 through Y31 for each of the 31 tiles, and then we have a latent hidden activity variable for each 5 base pair increment. So we have 59 5 base pair increments here, H1 through H59. And then what we can do is we can say Y1 here depends on H1 through H29, Y2 depends on H2 through H30, and so forth. So each Y variable depends on the H variables it overlaps. And then we make certain modeling assumptions. We assume that the Y variables are normally distributed with their mean depending on the mean of the incoming H variables at a variance that's set to the empirical variance of observed data. And then we assume that the H variables are also normally distributed with their mean set to the empirical mean of the observed data. And then we have a free variance parameter which controls the overall smoothness of the inference. In practice, what we do is we set it to two different values, a loose and tight value, and after our inferences, we do a z-score transformation on the inferred hidden variables, and then average their outputs. And just to note that The inference here can be done exactly because all these variables are normal distributions. So this becomes a big multivariate normal, and we can do inference exactly when we have multivariate normal distributions. So then what you're seeing here is our inferences at 60 different locations. I'm showing you the inferences for the HepG2 experiments based on, of all locations that we tested that had this specific regulatory motif predicted by the centipede method in red. So this is GAB-PA. In blue are other motifs predicted by the centipede method. In black is our overall inference. So you can notice that it's almost, it's usually above average. Sometimes it peaks over that regulatory motif. And now this especially contrasts if we look at another motif, in this case NRSF, which is a known repressive motif in red. And you can quite often see this dip of activity under where that NRSF motif is sitting. And again, we made this inference independent of knowing the NRSF motif or the specific DNA sequence. And we can do more systematic global analyses as well. So what I'm showing you now are examples of a few of these. So on the x-axis, I'm showing you the average HepG2 regulation score, and on the y-axis, K562. And each dot corresponds to a regulatory motif from a compendium, which was called just based on the DNA sequence. And you can see most dots sort of have consistent repressive activity or activating activity in both cell types. And this is an aggregation plot of specific motifs, like the ETS motif. The REST motif or NRSF was the most repressive in both cell types. One interesting thing that came out of this, the motifs that had the strongest activating activity in both cell types corresponds to this TENMER, which has come up in a number of contexts as being one of the most evolutionarily conserved motifs and near some of the highest expressed genes in vivo. But there's still some debate in the literature about what transcription factor actually binds this motif. And it was discovered in 5 different ENCODE ChIP-seq experiments de novo for different transcription factors. And then if we look off diagonal, that's where we can see some cell type specific activators and even repressors. So for example, HNF motif is activated specifically in this liver-related cell type, while GATA has activity specifically in K562. And one interesting thing here was this RFX family had repressive activity specifically in HepG2 cells. So that was looking at the average activity. One question that comes up is, are some motifs working as an activator and a repressor in the same cell type, which could get lost if we're just looking at the average? So one analysis we did here was looking, do we see an enrichment for these motifs above an activation threshold or below a repression threshold? And what we saw were a substantial number of motifs that were enriched based on below the repressive threshold and a large number above the activating threshold. And then if we zoom in here and look at motifs that were enriched for both these dual motifs, both having an activation and a repressive role in the same cell type, we only found a few examples, and most of them were from this MAF family of motifs. And then we also looked at other types of sequence elements, for example, repetitive elements, and turned out that in the HepG2 experiments, some of the most activating sequences were a certain type of LTR repeat called a ERV1 family. But this wasn't true in general, that repeat elements had high activity. For example, line was both depleted among the most activating and repressive motifs. And then we could also go back to our original chromatin state design here of selecting the regions and show that locations that we predicted as a promoter or enhancer had on average the highest activating activity, and then other states like a weaker enhancer had also above average activity, but not quite as high as a more active enhancer. And then the last analysis here that we did was we looked at, for each motif, its expected activity based on what chromatin states it was falling in. So if a motif, for example, fell into active enhancer and promoter annotated regions of the genome, we would expect it to have a high average activity just based on its chromatin context. And then we could also compute what's its actual observed activity, where we took those specific instances of the motif and then directly computed its average score based on this assay over those instances. So in general, there's a correlation between this expected and observed, but what's interesting is when we look at some of these outliers. So for example, this RFX family of motifs had, on average, among the highest expected activity, but also among the lowest actual observed activity. So in this case, the interpretation of this is this specific motif is effectively working as a staller or an attenuator motif. So in these active elements, so it's sort of working against the trend and sort of holding things back from being sort of completely active. And this had been noted for in like one specific enhancer, but here, using this high throughput technology, we can capture this trend more globally. And there's other examples like P53 sort of well outperforms its expected activity based on the chromatin context. So you can have P53 in locations that aren't specifically marked by these active enhancer promoter states, but are highly active in this assay if they have a P53 binding site. And then we saw the opposite for rest down here. Okay. Maybe if there's questions on that part, I could answer before I move to the next part, or there'll be a chance at the end. Yeah.
Sorry to bother, but okay, so I have two questions.
Sure.
Would you like to finish your talk and then see if you have enough time for my questions?
Okay, if you think they might be long questions or...
So actually, one is just the previous slide, if you don't mind.
Yeah.
Yeah. So I was wondering about these outliers. Could you associate some biological meaning to these? I mean, they were inferred, of course, probabilistically by the model, correct?
Yeah, I mean, so like the RFX one, I mean, there were specific examples in the literature about this one, but it was done at sort of these very small scale experiments. It was basically looking at one location. Here we can more statistically make statements about this.
And by the chromatin type, you said something about the expected?
Yeah, so the expected activity here is based on like the chromatin environment. So here I was saying if that motif was an enhancer or a promoter, you would expect to have higher activity, just because on average